Hello, dear students. Myself Sunita. Welcome to this session. In today's session, we shall be discussing about the next chapter, Chapter 2, Biological Classification. What exactly are we going to learn in biological classification? The word says biological, that means about the living organisms and its classification. So in the first chapter, you have learned what exactly is classification and what is the importance of classification. In this chapter, you will be learning the classification system which was proposed by various scientists according to their convenience, and some system of classification has been definitely made an remarkable achievement by the scientists which have been accepted universally. So in biological classification, we'll be understanding about the 2 kingdom, 3 kingdom, 4 kingdom and 5 kingdom classification. And also we'll be discussing in detail about the 5 kingdom classification, which is including the Monera, Protista, Mycota, Plantae, and kingdom Animaliae. So we shall look into our today's session with introduction to classification, and later on we'll be going through the kingdom Monera. Right. So as I told you, classification plays a very important role. So classification has been given by so many scientists, but the earliest classification was given by Aristotle. As we know, Aristotle is known as father of zoology, and he's also known as father of biology. So his system of classification, which he used, was ancient system of classification. And this system included, or the classification was done simply based on morphological characters. We all know what is morphology. Morphology is nothing but the external appearance. Morphology is nothing but external appearance. Okay, so just looking at the plant, he divided the plants into trees, shrubs, and herbs. So as he was a zoologist, he also divided the animals into 2 groups based on the presence of the blood. So into 2 groups, one with the presence of the red blood, with organisms with red blood cells or red blood, and the other one without red blood. Without red blood. So he called these organisms with the red blood as Enema, and without the red blood as Anema. Okay, so with the red blood, he called it as Enema, and without the red blood, he called these group of animals as Anema. Right? So this classification was given by Aristotle just based on the morphological feature with regard to the plants, and based on the presence or absence of the blood, he divided them into Enema and Anema. Then came the further classification, which was done by the father of taxonomy, that is Linnaeus. So according to Linnaeus' system of classification, the living world, he classified the living world into 2 kingdoms. So he called these 2 kingdoms as kingdom Plantae and then kingdom Animaliae, which we have learned in the first chapter. So this system of classification was recent in the sense recent to that of Aristotle, but it is also an ancient system of classification. So here he improvised his classification, and he also divided the living world, whole world, the living organism into 2 groups, that is plants and animals. Linnaeus system of classification also didn't give an appropriate classification since he did not include so many things, so many characters. So what are the characters which he did not introduce to the system of classification was, firstly, he did not distinguish between eukaryotic and prokaryotic organism, which is the most important for the classification. We know that prokaryotic organisms are those which are very having less number of characteristic feature, which do not have membrane bound organelles. Okay, and there are other properties also which do not possess in case of prokaryotic organisms. There is no distinct nucleus as well. Whereas in case of eukaryotic organism, U means true, pro means it is early or it was first, U means later, or it is also called as next, or it is called as true carry-on. The word carry-on here refers to nucleus. The word carry-on refers to nucleus. So these group of organisms, eukaryotes, they had true nucleus with the nuclear membrane. Okay, and also he did not distinguish between the unicellular and multicellular organism. That was the second demerit of his classification, which was not accepted. So he did not distinguish between unicellular and multicellular organism. He just dumped everything into plant and animal kingdom. So he did not differentiate between this is unicellular, this is multicellular. That was his second demerit. And also he did not distinguish between the photosynthetic organisms, that is green algae, and non-photosynthetic organisms like fungi. So green algae and fungi, they were both included under kingdom Plantae. So he did not differentiate between photosynthetic green algae and non-photosynthetic fungi. So since these were the demerits of his classification, so Linnaeus system of classification, that is two kingdom classification, was not accepted, right? Then apart from this, to understand the system of classification, there were very other groups which were present in kingdom Plantae, which Linnaeus included, like bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, angiosperms. Everything was considered to be under Plantae. But for not considering the points here, further the classification system was enhanced by few more scientists. There were two important criteria why two kingdom classification failed as such. One is, so many organisms here, especially bacteria, blue-green algae and all, the cell wall is different. The composition of the cell wall goes on varying in these organisms, right? And one more thing is that the cells may be unicellular or multicellular, as I told you earlier. So unicellular and multicellular organisms were grouped together along with the heterotrophs and autotroph organisms. So because of all the criteria which were not accepted, so there came up the next system of classification, that is three kingdom system of classification. The three kingdom system of classification was little enhanced by a German naturalist known as Ernest Haeckel in the year 1866. So he created one more kingdom along with the kingdom Plantae, which was already given by the Linnaeus. He included kingdom Plantae and kingdom Animaliae in his system and also introduced one more kingdom. And he named that kingdom, the third kingdom, as kingdom Protista. So kingdom Protista included unicellular protozoans and algae. This again did not form a merit for the system of classification because once again the same mistake was repeated even by Ernest Haeckel. So Ernest Haeckel did not include or did not distinguish between prokaryotes and eukaryotic organisms. He simply said that all unicellular organisms, protozoa and algae, they all are into one group, and he henceforth called the kingdom as Protista. So third kingdom classification or three kingdom classification also was failed, and henceforth came up the fourth kingdom classification. The fourth kingdom classification was given by Copeland, and he proposed one more kingdom along with three more other kingdoms which were already given by Ernest Haeckel and Linnaeus. So already there are now three kingdoms which included kingdom Animalia, kingdom Plantae, and also Protista. Now came up one more kingdom, which was called as Kingdom Monera, Co-plant included the organisms under Monera, which were prokaryotic, that is bacteria, blue-green algae. They all belong to the kingdom called as Monera. So from Plantae, now the bacteria and blue-green algae were separated and then put into one more new kingdom, which was called as Monera. But one of the demerit again, which Co-plant didn't act on, or which became a demerit for his classification, was he did not separate prokaryotic and eukaryotic organisms. Along with that, he included other organisms as well in his kingdom of classification. So like autotrophic organisms and heterotrophs were not again distinguished or differentiated. So once again, that was a mistake where everything was included under the same kingdom, Monera. Later came one more system of classification after 4 kingdom, that is the 5 kingdom system of classification. NCERT textbook, what has been given is regarding the 2 kingdom classification and the 5 kingdom classification. 5 kingdom classification is the most accepted system of classification, which was proposed by Robert H. Whittaker. So Robert Whittaker, he proposed this in the year 1969. The year has to be remembered here because his system of classification was a remarkable one. Why? Because he took all the demerits which was not considered to be a merit in his in the earlier system of classification, and all the characteristic features, which includes heterotrophic, autotrophic, prokaryotic, eukaryotic, unicellular, multicellular, all the characters were included, and then the classification system was defined. So henceforth, Whittaker's system of classification was the most accepted system of classification at the universal level. So his system of classification was called as 5 kingdom system of classification. Already there are 4 kingdoms, that is Animaliae, Plantae, given by Linnaeus. Ernest Haeckel gave Protista. Co-plant gave Monera. He included one more kingdom called as Kingdom Fungi, or it is also called as Kingdom Mycota. Kingdom Mycota included the group of organisms which are also called as fungi. So according to his classification, the main criteria which he took to classify the organisms or the characters included the cell structure, the thallus organization, that is how the plant body is organized, then the mode of nutrition, then reproduction, and phylogenetic relationship. So he included even the phylogenetic relationship along with other characters, which gives an link between one organism to another organism or one kingdom to another kingdom at the taxon level. So henceforth, 5 kingdom classification was most accepted system of classification, which was proposed by R.H. Whittaker. So here, all prokaryotic organisms were grouped together as Monera, and unicellular eukaryotic organisms were placed under Protista. Kingdom Protista included Chlamydomonas, chlorella, which was earlier placed in Plantae because they had only cell wall as one criteria, but later they were grouped under Protistans. Paramecium amoeba, earlier they were placed under animal kingdom. Now it has been placed under Kingdom Protista. Since they lack cell wall, it was grouped under Animaliae, But since they are unicellular organisms, they have been grouped unicellular eukaryotic organisms. Henceforth, they are grouped under Protista. So because of so many merits, this system of classification plays a very important role. So Robert Harding Whittaker, who gave this classification system, and he also said the relationship, evolutionary relationship between Monera. From Monera originated Protistans. From that originated Plantae, Animalia, and fungi. So he got to know that the Monera showed the least simpler characters. So the organization is very simple. Henceforth, he rearranged the whole system of classification into first is Monera, then comes Protista, then comes fungi, then comes Plantae, and then Animalia. So accordingly, he classified from the simplest to the complex organization, right? So now we look into this chart, which was given by R.H. Whittaker, taking into consideration so many characters like cell type, cell wall, nuclear membrane, body organization, and mode of nutrition. Coming to Monera, so we can look into the table. It is a most important NCERT table. Number of MCQ questions will appear out of this table. So kindly focus and note it down. Monera include cell type as we are talking about the prokaryotic or eukaryotic cell. So as we know, Monera includes the prokaryotic cell. That is, prokaryotic means the nucleus prokaryon. Karyon refers to the nucleus, as I told you earlier. That means they are primitive in nature. That is without the nuclear membrane. So prokaryotic organisms are those organisms without the distinct nucleus. Without distinct nucleus. That is without the nuclear membrane. Then Protista, fungi, Plantae, and Animalia, they all come under eukaryotes. So all eukaryotes were put together starting from Protista to Animalia. Then comes the next character, that is cell wall. So as I told you earlier, the cell wall composition goes on varying from one group of organism to another group of organism. As we go ahead with each and every kingdom with regard to the animals, we'll understand much more about the cell wall composition. So here we can understand that they are made up of non-cellulose. Generally, we know that the plant cell is made up of cellulose, but here they are made up of non-cellulose, a polysaccharide. Polysaccharide is a sugar and along with an amino acid. Amino acid serves as a units of proteins, okay? In case of protistans, if the cell wall is present, but they are present only in some, and it goes on varying. They may be made up of silica, they may be made up of proteins, they may be made up of even calcium. Okay, it varies. Or it may be made up of cellulose as well. So that goes on varying. Then coming to the fungi. Fungi, the cell wall is present. In all these, the cell wall is present. But in case of fungi also, they are not made up of cellulose, but they are made up of a polysaccharide, which is called as chitin. In case of plant, we know that the cell wall is present and it is made up of cellulose. Along with cellulose, it depends on the plant cell whether it is made up of the other types like hemicellulose, pectin, lignin, etc. And we know that animals are differentiated because they do not have any cell wall. So cell wall is completely absent in case of in case of animal cell, whereas the cell membrane constitutes the outermost lining in case of animal cell. Coming to the nuclear membrane. So nuclear membrane is absent. That is why they are called as prokaryotes. Whereas in case of eukaryotes, they are present in all the groups. Then next one is body organization. What are they made up of? What exactly is the body made up of? Since you know that Monera is the least in its organization, they're just made up of cells, which forms the basis of life, the unit of life, that is nothing but cell. Even in Protista, they are all unicellular organisms. Monera and Protista, majority of these organisms are unicellular, which are made up of single cell. Starting from fungi to the animal, they are all made up of multicellular organisms. So multicellular cells together may constitute the tissues which are loosely packed. In case of plants, the tissues may be constituting the organ. In case of animals, they may be cellular level, tissue level, organ or organ system level, which you'll be learning in detail in plant kingdom and animal kingdom, which is in 3rd and 4th chapter. Lastly, about the mode of nutrition. So the mode of nutrition is basically two types. One is the autotrophic mode. Some of them may be autotrophs, and some organisms may be heterotrophs. Auto means they can prepare their own food on their own. Hetero means they are dependent on organisms for food. Again, the autotrophs may be divided into two types based on what type of food they are going to obtain from what source of energy. So they may be chemoautotrophs or chemosynthetic, or they are also called as chemoautotrophs. And the other group is photosynthetic or photoautotrophs or photosynthetic. So depends on the energy. Photosynthetic, photo means light. They are going to derive the energy from the sunlight and then they are going to utilize the food, or they are going to prepare the food, synthesize the food. Whereas chemoautotrophs, they are going to synthesize using the chemicals, inorganic chemicals, right? Then heterotrophs, they are purely dependent on the other group of organisms like autotrophs, and they may be saprophytes or parasites. If they are saprophytic in nature, they are going to depend on dead and decayed organisms. They are going to depend on dead and decayed organisms. If they are parasites, if they are going to be parasite, then they will be depending on the host body. They may be present on the host. They are called as ectoparasites, or they may be present inside the host body. They are called as endoparasites. So this is regarding the nutrition. Now let us look into what type of nutrition do they follow. Monera, generally you have all the types. You have both autotrophs as well as heterotrophs. In autotrophs, both photosynthetic and chemosynthetic is present. In heterotrophs, they are both saprophytic as well as parasitic. In protistans, majority of them are autotrophs and some of them are heterotrophs. If they are autotrophs, majorly they are photoautotrophs. In case of fungi, they are purely heterotrophic organisms. They may be either saprophytic or they may be parasitic. Then comes plantae. We know that plants can prepare their own food using the process called as photosynthesis. They are purely dependent on sunlight. There are heterotrophic organisms. In case of Animalia, you know that they are dependent directly or indirectly on the plants. They are holozoic, they are saprophytic. So holozoic is a mode of taking food through the process called as ingestion. Eating is called as holozoic. And the other mode is they are going to depend on the dead organisms. Okay, so this is regarding the 5 kingdom classification, and we understood the overall characteristic features for the different for classification done by R.H. Whittaker. So now let's go ahead and understand one more system. So earlier in the year 1977, after R.H. Whittaker proposed his 5 kingdom classification, came up one more system of classification called as 6 kingdom classification. Came up called as 6 kingdom classification. 6 kingdom classification, already 5 kingdoms, you know, starting from Monera to Animalia. So there comes one more system of classification called as 6 kingdom classification. So this 6 kingdom classification was given by Carl Woese in the year 1977. He did not call it as kingdom, but rather called it as domains. So till now we have learned the hierarchy as, if you remember the formula KPC of GS, that is kingdom, phylum, class, order, family, genus, and species. So above the kingdom level comes one more category called as domain. So group of kingdoms called as domain. So Carl Oers divided into three important domains, which included archaea, bacteria, and eukarya. So archaea included all the organisms which are present in the harsh habitats like extremophils, methanogens, halophils, thermoacidophils. Under bacteria, he included cyanobacteria, eubacteria, beneficial and pathogenic bacteria. Majority of them are heterotrophs. And then the last group, eukaryotes, were grouped under one single domain called as eukarya, which included protista, fungi, plants, and animals. So we know that bacteria are grouped under, according to the Whittaker system, into Monera, and protista, fungi, plants, animals, they all belong to eukaryote, that is eukarya domain, but they all are kingdom under R.H. Whittaker. So protista is another kingdom starting from Monera, protista, fungi, plantae, and animalia. Now the fifth kingdom which we have to include is kingdom archaea, which includes archaebacteria. So archaebacteria was introduced by Carl Woese. So this group of organisms are bacteria again, but they are going to be present in harsh habitats, like they are going to be present in thermal hot springs. They are going to be present in saline conditions. They are going to be present in the polar regions. So where there is extreme of habitat, those organisms, bacteria which are present there, they are called as archaebacteria, okay, which include extremophils. They all are extremophils. Together we call it as extremophils, which are going to be present in harsh habitats like methanogens, halophiles, thermoacidophiles are the three important groups. So this is the six kingdom classification which was proposed by Carl Woese. Instead of calling as kingdom, he also named it as three domains, where archaebacteria grouped under one domain, bacteria were one domain, and the rest all other eukaryotic organisms which were present starting from protista to animalia were grouped under one domain. So let's move on to our first kingdom, that is Monera. So in kingdom Monera, what are we going to learn? So kingdom Monera includes the sole genus or sole organisms, that is bacteria. So we'll be learning about different types of bacteria. Since we are going to follow the Whittaker system of classification and not Carl Woese system of classification, henceforth the kingdom Monera will be the having the very less characteristic features, and that will be the first kingdom, which includes bacteria, cyanobacteria, archaebacteria, and mycoplasmas. So we'll be understanding about all these in today's session. Kingdom Monera, as I told you, it includes the sole members called as bacteria. So bacteria are the group of organisms which are unicellular in nature. They are unicellular organisms, prokaryotic in nature. Prokaryotic in nature. And bacteria, if we talk about the occurrence, where is it present? They are present everywhere. They are present everywhere. So they are found in abundance and they are found everywhere. Let's take example of water. They are present there. They are present in soil. They are present in air. They are present in and around us. So they are present within us. They are present everywhere. And they are also present in extremes of habitat. So they are also extremophils, which we call it as extremophils. They are present in extremes of habitat. And apart from that, we also should know that they may be present on the host body or within the host body. Host in the sense, it may be human or it may be any organism, which they may live upon them or they may be inside them. So bacteria, they are grouped or classified based on the shape into four types. That is four types. So this is a typical bacterial structure. So there are four types. One is the coccus. Then comes bacillus. Then comes Vibrio. And lastly, the spirillum. Coccus is spherical shape. Bacillus is rod shaped. Vibrio is comma shaped. And spirillum is spiral shaped. So in plural, coccus is called as cocci, bacillus is called as bacilli, Vibrio is called as vibrio, and spirillum is called as spirilla. So these are the bacteria based on the shape. The picture which is shown here is a bacillus. It's a rod-shaped bacteria. And typical bacterial cell has the cell wall. The typical bacterial cell has a cell wall. It has the plasma membrane as the innermost layer. The cell wall in case of these bacteria is made up of a specialized sugar molecule which is called as peptidoglycan. Along with other components. And inner to the plasma membrane is the cytoplasm. The cytoplasm is having mesosomes, the ribosomes, and the DNA, which is present as a genetic material. The nucleoid of the cell is not covered by or having the distinct nuclear membrane, so it is called as nucleoid. And also it has a locomotive structure which is called as flagellum. Then coming to the mode of nutrition. When we talk about the mode of nutrition, it may be both autotrophic or it may be heterotrophic. Autotrophs or they may be heterotrophs. In autotrophs, there are two types as we discussed. They may be photoautotrophs or photosynthetic, and they may be chemoautotrophs or chemosynthetic. Then comes heterotrophs. They may be parasitic in nature or they may be saprophytic in nature. Right? Now, just look into the shapes of bacteria. So you can see the spiral shaped, rod shaped. I'm sorry, it's spherical shaped. Spherical, rod shaped, spiral shaped, and comma shaped. Generally, the spherical shape do not have flagella. So the arrangement of cocci and bacilli, like that, it goes on varying. If they are single, they are called as monococcus. If they are in two, they are called as diplococcus. If they are in bunch, they are called as staphylococcus. If they are in the form of chain, they are called as streptococcus. If they are present in the group, they are called as sarcina. And if they are in four, they are called as tetroid. This is applicable even to the rod-shaped bacteria, that is bacillus. They may be monobacillus, diplobacillus, like that it goes on. Coming to the classification of Monera, kingdom Monera, which is classified into Archaebacteria and Eubacteria. first we'll be learning what exactly is Archaebacteria, and later we'll go ahead with Eubacteria. The word Archaea represents ancient bacteria, ancient group of bacteria. So when we look into the habitat of these bacteria, basically they'll be present in harsh habitats. As I mentioned you earlier, harsh habitats include the extremes of conditions like salty areas, or they may be present in marshy areas, or they may be present in hot springs. Right? So the organisms which are present are having the cell wall different from those of Eubacteria. So that is why, because of the cell wall composition, since there are different composition of the cell wall, these organisms are able to survive in extremes of habitat when compared to the other bacteria which are generally present all over. So you can see here the bacteria, especially the Archaebacteria, they have something called as ether chains or ether links, whereas in case of Eubacteria, they are going to be having ester links. And here the composition is there is branched and there is unbranched. The chemical composition of the cell wall is what will differ in the bacteria, Archaebacteria and Eubacteria. That is why these bacteria can live in extremes of habitat because of the cell wall nature. Then some of the examples which we have to study is methanogens. So as I told you, methanogens are those which are going to survive in marshy habitats. Halophiles are those which are going to survive in salty areas like seas and ocean. And thermoacidophiles are those which are going to survive in hot springs. So all together, methanogens, halophiles, and thermoacidophiles, they are called as extremophils. And along with that, methanogens, not only they are present in marshy areas, they're going to be present in the gut, that is the alimentary canal of the animals, especially like cows and buffaloes, the domestic animals. Since they are present inside the cows and buffaloes, they help during the process of digestion. And in these organisms, they are going to produce a methane gas. They are going to help in breakdown of organic substances to produce the gas, which is called as methane gas. And that methane gas is used as a biogas. Methane is used as biogas, which is obtained from the dung of these animals. So they are going to be economically important as well. Okay, so this is regarding Archaebacteria. Now we shall look into Eubacteria. So Eubacteria, they are also called as true bacteria. The word Eu means true. Why are they called as true bacteria? Because when compared to Archaebacteria, they are going to survive in different habitats. In and around what we see, the bacteria are going to be called as Eubacteria. Whether they are soil bacteria, they are going to be airborne bacteria, or they may be even present in the water. So they are all called as true bacteria, or they may be even terrestrial form as well. Okay, so these bacteria, they have rigid cell wall, and this cell wall is made up of peptidoglycan. The cell wall material of bacteria is called as peptidoglycan. And majority of bacteria, they are motile in nature, because we know that these bacteria carry a thread-like structure. So this thread-like structure is known as flagellum, which is going to be serving for the process of locomotion. The coccus bacteria is, since it does not have flagellum, it's not stagnant, but it can move through the water currents or air currents. Okay, gliding movement, we call it as gliding movement. They just glide over the surface. Then in this eubacteria, we are going to understand based on the mode of nutrition, we classify the bacteria as photoautotrophs, and the other group is chemoautotrophs. They are autotrophic bacteria. One more group is heterotrophic bacteria, which we'll be discussing after this. So first we'll understand about the photoautotrophs. Photoautotrophs are also called as blue-green algae. One of the example of photoautotrophs is blue-green algae, which in turn is called as cyanobacteria. So cyanobacteria is also called as blue-green algae. So as you can see in this picture, they are the bacteria which are generally unicellular. They are unicellular, or they may be in the form of colonies, which we call it as colonial, or some of them are filamentous form. They may be present in both fresh water, just like ponds, lakes, etc. They may be present in freshwater forms or marine as well. They may be even marine organisms. And apart from that, they can also be terrestrial forms, that is present in the soil. So when we have to take a typical example of these eubacteria, especially the photoautotrophic bacteria or cyanobacteria, which is also called as blue-green algae. So typical examples taken here is nostoc. So nostoc, as you see in the picture, is a filamentous blue-green algae. It's a filament. So what you're seeing here, these are the filaments. Okay, they are unicellular. These are all filaments. Instead of cell, we call them as filaments. Many filaments are joined together, and the structure is called as filamentous structure. The diagram is very important. In each filament, if we have to see, there is so much amount of cytoplasm, and also the pigments are present here, which gives the coloration to these organisms, which appear to be bluish green in color. Surrounding these organisms, there is a sheath called as mucilaginous sheath, which protects the organism. And in few filaments, the colored pigments or the cytoplasm is not present. Instead, it forms a hardened structure, which is called as heterocyst. So these heterocysts are nothing but the specialized cells. Heterocyst are specialized cells to perform function of the function of nitrogen fixation. So the typical function of this heterocyst is to perform nitrogen fixation, and they are of great importance because they fix atmospheric nitrogen and they play a very important role. There is one more example other than nostoc. This picture is nostoc, and one more example is there called as Anabina. A-N-A-B-A-E-N-A, Anabina. So nostoc and Anabina are the two typical examples of cyanobacteria or blue-green algae. So this is regarding the photosynthetic autotrophs. Now let's move on to the next group, that is chemosynthetic autotrophic bacteria. So photoautotrophs, they're going to synthesize food by utilizing the sunlight, whereas the chemoautotrophs, as I told you, they're going to utilize or oxidize the inorganic substances to form into organic substances, and that is how they're going to produce the energy. So this is cyanobacteria. So generally cyanobacteria, wherever they are present in the water bodies, they are going to pollute the water bodies. Okay, they are generally polluting the water bodies. They form the water blooms. We call it as water blooms, which pollute the water bodies. Some of them may be harmful, some of them are not. Some of them release the toxins. So that is how they create chaos in the water bodies. Next, chemoautotrophs. Chemoautotrophs are the bacteria which oxidize various inorganic substances. The inorganic substances may include nitrates, nitrites, and ammonia, which are nothing but the compositions of nitrogen, and use the released energy for their ATP production. ATP is the energy molecule which is essential for functioning of these bacteria. So henceforth, to produce ATP, they have to oxidize, they have to undergo the process of oxidation. Inorganic substances are going to oxidize, and later the food material, whatever has been produced, will release energy in the form of ATP. And that ATP is utilized for the activities by the bacteria. And apart from these, chemoautotrophs play a very important role in recycling nutrients. They are utilized in biogeochemical cycles, which are involved in the processes of the nutrient cycling, biogeochemical cycles, which are essential for nutrient cycling, recycling, like nitrogen cycle, phosphorus cycle, iron cycle, and sulfur cycle. So chemoautotrophs include bacteria which are going to fix the nitrogen. They're called as nitrifying bacteria, which include Nitrosomonas and Nitrobacter, which converts atmospheric nitrogen into nitrates and nitrates into nitrites. And there are few other chemoautotrophs like sulfur, which is used as an oxidizing substance, and also iron bacteria, where iron is used as an oxidizing substance. So this is regarding the chemoautotrophs. Next, going ahead with the heterotrophic bacteria. Heterotrophic bacteria, when compared to the autotrophic bacteria, are abundant in nature. The number of organisms when compared to archaebacteria, when compared to the bacteria that is photoautotrophic bacteria, heterotrophic bacteria, they are present in abundance. Majority of them here are decomposers. So henceforth, the number of these organisms will definitely increase. So what is the importance of heterotrophic bacteria? They are of high economic importance. They are the one which are going to make curd from milk, which we call it as curdling of milk. Major organism here is lactobacillus in the milk, right? And some of them are going to produce, they are important in medicinal field, like for the production of antibiotics. Some of them, they are going to fix nitrogen, especially in the legume roots. One example is rhizobium. Some are pathogenic in nature. They are not only pathogens for human beings, but also they are going to be pathogenic for crops and farm animals and even pet animals. Then they are going to, these are some of the useful, bacteria. And there are even harmful bacteria, which include the disease causing bacteria, like they are going to cause cholera, they're going to cause typhoid, they're going to cause tetanus, they're going to cause even harm to the plants, especially the citrus plants, which is called a citrus canker. So there are a lot of other bacterial diseases which are going to be caused by these heterotrophic bacteria in both plants as well as in animals as well as in human beings. Then comes the overall reproduction in bacteria. So basically, the reproduction is of two types. One is bacteria undergo the reproduction. We can say it as asexual reproduction by binary fission. Binary fission means a bacteria is able to divide itself into two, and then equal distribution of genetic material and the cytoplasm will be split into two parts, and that process is called as binary fission. A single bacteria is able to divide itself into two, so forming the progenies where the genetic material is equal. You can see the genetic material here, and this type of process of splitting is called as binary fission. Bi means two, fission is nothing but splitting into two. So this is a asexual mode in case of bacteria. But during unfavorable conditions, the bacteria are not able to produce any type of multiplication in their number. So henceforth, they remain to be resistant and they form the spores. Okay, they are going to form different spores. When they undergo the sexual reproduction, they are of three types, that is due to genetic recombination. So first one is transformation, conjugation, and transduction. In detail, you will be learning this in the second year topics. So transformation is a type where the genetic material will be passed on from one bacterium to another bacterium. Conjugation is a technique where the conjugation bridge is going to be formed between the bacterium, and that is how the genetic material will be passed on. Transduction is by using an virus which affects bacterium, which is called as bacteriophages. So by bacteriophages, the genetic material is passed on from one bacterium to another bacterium. You can see here the bacteriophages are going to attach themselves, and later the transduction is going to happen from one bacterium to another bacterium. Okay, so these are the three important methods where genetic recombination or transfer of genetic material is going to take place from one bacterium to another bacterium. So this is the means of sexual reproduction. Next, the last group of kingdom Monera includes mycoplasmas. Mycoplasmas are the group of specialized organisms which come under bacteria. They have three important things to be noted here. So this is the microscopic picture, what you're seeing on your right side, and this is the diagrammatic picture of mycoplasma. So mycoplasma are those bacteria which are going to be causing a disease called as pleuropneumonia-like organism, that is PPLO. PPLO refers to as pleuropneumonia-like organisms. Pleuropneumonia-like organisms is PPLO. And the speciality of these organisms is that they lack cell wall. Okay, so in all other bacteria, the cell wall plays a very important role, but in case of mycoplasmas, they are going to be lacking cell wall. And then they are the smallest living cells known. So it has been discovered that they are very small in nature, and bacteria itself is very small, and they are microscopic in nature, and mycoplasmas are even more smaller when compared to the other types of bacteria. They are the smallest living known cells. They are unicellular. They are the smallest living known cells. Then they can survive without oxygen. Generally, some of the bacteria can survive without oxygen. Some cannot survive without oxygen. They may be aerobic bacteria or they may be anaerobic bacteria. But these mycoplasmas can survive without oxygen. They can survive in anaerobic conditions. Aerobic conditions. And they are highly pathogenic in nature. They are pathogenic for both plants as well as animals. Okay, they are pathogenic for both plants as well as animals. So this is regarding Monera, kingdom Monera. Now we'll look into some of the questions related to the topic which we have discussed. The first question is, all eukaryotic unicellular organisms belong to? We know that all eukaryotic unicellular organisms. Basically, unicellular organisms, the group, that is the kingdom where unicellular organisms belong to, is only Monera and Protista, but eukaryotic organisms are belonging to Protista, whereas prokaryotic organisms are belonging to Monera. Henceforth, the option here is Protista. Second question, the five kingdom classification was proposed by, we know that Coriolanus Linnaeus proposed two kingdom classification, Ernest Haeckel proposed three kingdom classification, Copeland proposed four kingdom classification, and R.H. Whittaker proposed five kingdom classification. Henceforth, the option is A. Next question. Organisms living in salty areas are called as, we know that methanogens are those bacteria which are going to be living in marshy habitats and in the ruminant or in the gut region of the cattle and buffaloes. Halophiles are those organisms, those bacteria which are going to be surviving in the salty areas. Halo refers to salty. Okay, philes refer to bacteria. So these organisms which are surviving in the salty areas, they are called as halophiles. Thermocidophiles are the one which are going to be surviving in hot springs, thermal hot springs. Thermo is temperature, so where they are hot. Acidophiles refer to high temperature, so they are called as thermoacidophiles. Then blue-green algae are classified under, we know that blue-green algae, eubacteria and archaebacteria, they all belong to kingdom Monera. Even though earlier they were considered to be under Plantae, but according to the Whittaker system of classification, it has to be grouped under Monera. Next question. The smallest living cell, mycoplasma. So what exactly are the correct characters we need to identify here? Completely lack a cell wall. Second option, cannot survive without oxygen. Third option, are pathogenic to plants and animals. Fourth option, do not reproduce by fission. So we know that completely lacks cell wall. And also we know that they cannot survive without oxygen. They are pathogenic in nature to plants and animals. All the three statements are correct. So you can include one more here. So option 1, 2, and 3, all three are correct. Okay. Next question. In five kingdom classification, which single kingdom includes blue-green algae, nitrogen-fixing bacteria, and methanogenic archaebacteria? So as we know that all the bacteria are grouped under one single kingdom, that is Monera, which is the sole genus or sole members of kingdom Monera. So henceforth, the option will be option B. Next question. Which of the following characters served as a criteria for five kingdom system of classification as used by Whittaker? Is it cell structure, body organization and mode of nutrition, reproduction and phylogenetic relationship, or all the above, all of above? So we know that cell structure was considered, body organization was considered, mode of nutrition was considered, reproduction was also taken into consideration, and phylogenetic relationship was also considered. Henceforth, the option is all of the above. Next question. State whether the given statements are true or false. First statement, bacteria show both autotrophic and heterotrophic nature. Yes, it's true. It shows both autotrophic and heterotrophic nature. Some of the bacteria are autotrophic. They may be photoautotrophic or photosynthetic autotrophic or chemoautotropes or chemosynthetic autotropes. We know that they include both the means of nutrition. Next, heterotrophic nutrition involves obtaining ready-made organic nutrients from outside sources. So here we know that heterotrophic organisms are those which are depending on the other. They are going to oxidize the inorganic substances and then convert them into organic substances. So that is how they are going to obtain from different types of sources. So henceforth, all the three options are true. The correct option here is option C. Next one. Comma-shaped bacterial cell is called as what? Comma-shaped bacterial cell. We know that Vibrio is called as comma-shaped bacterial cell. Cocci is spherical. Spirillum is spiral. Bacilli is rod-shaped. So henceforth, comma-shaped bacterial cell is called as Vibrio. Last question. Most abundant bacteria are, so we know that most abundant bacteria belongs to, is it chemosynthetic, heterotrophic symbionts, heterotrophic decomposers, archaebacteria? So since we know that most abundant bacteria which are present is heterotrophic decomposers, henceforth the option is option C. Thank you, students. We'll meet you in the next video.